Hello, I am DK Lee, Chief Executive Officer of OliX. It has been approximately 1 year since we hosted our R&D Day on April 29th of last year. During this period, OliX has achieved several meaningful milestones and made significant progress. Today, I will begin by providing a high-level overview of these achievements. Following this, our Head of Research and Head of Business Development will present more detailed updates on our R&D and business development activities. I will jump into the main topics. The building shown on the screen is our new headquarters, located in Seongnam, approximately a 30-minute drive from Seoul, which we moved into in October 2024. Beginning in 2025, our workforce has gradually expanded, and we now have slightly over 100 employees. We continue to recruit highly qualified talent to further strengthen our organization. Of our total headcount, approximately 80 employees are based at our headquarters in Korea, while the remaining 20 are part of our U.S. subsidiary, OliX US. OliX US operates in 2 locations, Boston and San Diego. In San Diego, we have our chemistry lab, where a team of about 15 chemists is actively conducting research. In Boston, our C-level executives, along with teams in CMC, clinical, and alliance management, are based. I will briefly introduce our core technology, RNA interference, or siRNA technology, among gene silencing approaches based on oligonucleotides. This technology is widely regarded as both the most potent and the most well-validated. Since the phenomenon was first discovered in 1998, its significance was recognized with the Nobel Prize in Physiology or Medicine in 2006, just 8 years later. Subsequently, Elna Ilram, a pioneer in RNAi therapeutics, founded in 2001, received FDA approval for its first drug in 2018. As shown on the right side of the slide, RNAi therapeutics have since achieved an average of approximately 1 FDA approval per year, with a total of 8 products currently on the market. siRNA therapeutics offer a meaningful opportunity to target so-called undruggable areas that have been difficult to address with conventional approaches, effectively expanding the scope of drug development. In addition, as a platform technology that can be applied across multiple disease areas, they also provide strong scalability and long-term growth potential. A core advantage of this platform lies in its ability to achieve selective delivery to target organs such as the liver and adipose tissue. Once a delivery platform is established, multiple drug candidates can be rapidly generated in a short period of time simply by modifying the nucleotide sequence of the siRNA. Recently, OliX has branded its proprietary RNAi platform as OASIS, and is leveraging this platform to develop a range of innovative therapeutics. The OASIS platform is based on chemically modified asymmetric siRNA structures conjugated with various ligands to maximize delivery efficiency to target tissues. For example, when targeting the liver, we attach a ligand known as GalNAc, whereas for delivery to the skin, eye, or adipose tissue, lipid conjugates are used to enable efficient tissue-specific delivery. Furthermore, we are expanding the platform to additional target areas, including the central nervous system and muscle, by incorporating a broader range of ligand technologies, including antibodies, thereby continuing to extend the capabilities of the OASIS platform. Let me briefly summarize the key achievements OliX has made from our last R&D Day to today. I will highlight 4 main areas. First, the expansion of strategic partnerships. Second, securing a stable financial foundation. Third, strengthening our pipeline under the OliX 2.0 strategy announced in August last year. And finally, the successful establishment of both dual targeting and the dual organ targeting platform, which we are introducing for the first time today. First, on the expansion of strategic partnerships. As many of you are aware, we have been closely collaborating with Hansoh Pharmaceutical, one of the leading pharmaceutical companies in China, on multiple targets in cardiometabolic diseases. In addition, our MASH therapeutic candidate, OliX 702A, is partnered with Eli Lilly and is currently in the final stages of a Phase 1b clinical trial. We have also initiated our joint research with L'Oréal, which we were not able to discuss in detail previously due to confidentiality. This collaboration focuses on skin and hair regeneration, as well as longevity, and has been progressing very successfully to date. Notably, just last month, we achieved a significant milestone by securing an investment from Bold, the venture fund of the L'Oréal Group, marking Bold's first investment in a publicly listed Korean company. In addition, we have entered into a new framework agreement to further expand our existing collaboration. Further details will be provided in the BD session that follows. Next, I will address our efforts to secure a stable financial foundation. The day following our R&D day last year, we received a milestone payment from Hansoh Pharmaceutical upon the exercise of an option. Subsequently, in June, we entered into a co-development agreement with L'Oréal in the fields of skin and hair. In August 2025, we successfully raised 115 billion won through a CPS issuance from leading local and global institutional investors. This provided us with a solid financial base to further accelerate our R&D efforts. On the back of this strengthened foundation, we achieved several key milestones, including the successful completion of the Phase 1 clinical trial for OliX 301A in age-related macular degeneration, and the initiation of dosing in the Phase 1b and 2a clinical trial in Australia for OliX 104C, our hair loss program. Furthermore, in June of this year, we raised an additional 111 billion won from Bold, the venture fund of the L'Oréal Group, and US-based private equity firm Weiss Asset Management. Notably, this financing was conducted entirely through common equity, which we believe reflects strong market confidence in OliX's future value. In parallel, we also entered into a new framework agreement with L'Oréal to further expand our collaboration. Based on these achievements, OliX currently holds over 200 billion won in cash and cash equivalents. This strong financial position provides a solid foundation for us to steadily advance our technology development and pipeline expansion. Against this backdrop, let me also briefly touch on our market capitalization. At the time of last year's R&D Day, our market cap was approximately 960 billion won. It increased significantly at one point, reaching a peak of around 4 trillion won, but has since adjusted and is currently in the range of 2.5 to 2.6 trillion won. I share these figures not to highlight our achievements, but to provide context on our journey over the past year. While we have made meaningful progress both internally and externally, we are also very mindful of the recent share price performance. We sincerely appreciate the continued support and patience of our investors. Going forward, we will remain focused on delivering steady results and transparent communication with the aim of rebuilding and strengthening shareholder confidence over time. Next, I will discuss our OliX 2.0 pipeline. Based on our OASIS platform, we have historically focused on the liver and locally accessible tissues, such as the skin and ophthalmic areas. With the announcement of OliX 2.0 in August last year, we set a new goal to develop a platform capable of delivering siRNA to extrahepatic tissues, that is, major internal organs beyond the liver, and thereby expand the scope of siRNA therapeutics. Since then, we have made meaningful progress toward this objective. More specifically, OliX 2.0 is a strategy to enable effective delivery to a range of extrahepatic tissues, including adipose tissue, the central nervous system, kidney, and muscle, and to expand our pipeline accordingly. Based on this strategy, we are actively advancing a diverse set of programs. As a result of these efforts, a total of 4 programs have now entered global clinical development. In addition to the organs targeted under OliX 1.0, we continue to build new pipelines targeting previously inaccessible tissues. Our MASH therapeutic, OliX 702A, partnered with Eli Lilly, is currently in the final stage of a Phase 1b clinical trial in Australia. The first patient was dosed in June 2024, and the last patient visit was completed in May of this year. Accordingly, we expect to receive the clinical study report within this year. Due to our agreement with Lilly, we are limited in the level of detail we can disclose. However, what we can share is that, based on our NAFLD patient cohort data, we have observed best-in-class levels of liver fat reduction along with durable effects. Under the terms of the agreement, we expect our partner to lead subsequent clinical development. Moving to our platform applications in dermatology, next is our OASIS Skin program. Our most advanced pipeline in this area is OliX 104C, a treatment for hair loss, which is currently in Phase 1b and 2a clinical trials in Australia. We completed the Phase 1a study in January 2025 and initiated dosing in the Phase 1b study later that year. In the second half of this year, we plan to complete Phase 1b and proceed into Phase 2a, with the goal of completing Phase 2a by 2027. To date, no systemic adverse effects related to systemic exposure have been observed. In particular, no hormone-related changes, including those involving androgens, have been detected. We believe this represents a key point of differentiation from existing hair loss treatments. We have thus far observed a highly favorable systemic safety profile, and efficacy will be evaluated in the Phase 2a study. Separately, in addition to OliX 104C, we are also developing a cosmeceutical program under the codename OliX 104cc. This product was piloted last year under the brand name Uberna and field tested. We received very strong and enthusiastic feedback from users. Based on these results, we are planning an official market launch of Uberna in the second half of this year. Next, I will discuss our ophthalmology platform, OASIS Eye, OliX 301A, which targets both wet and dry age-related macular degeneration, has completed its Phase 1 clinical trial, and we plan to submit an application for a Phase 2a trial in the second half of this year. The Phase 1 study was completed in November last year, where we observed a highly favorable safety profile. In addition, despite being a Phase 1 study, we observed encouraging signals of vision gain in multiple wet AMD patients, as well as in a subset of dry AMD patients. Based on these results, we have decided to focus our Phase 2a trial on patients with geographic atrophy, a subtype of dry AMD. We plan to submit the relevant application in the near future. While detailed study design cannot be disclosed at this time due to regulatory requirements, the trial is expected to be conducted as a multi-regional study, enrolling approximately 80 patients with dry AMD. In parallel with these clinical efforts, we are actively engaged in discussions with multiple global partners across various strategic options. These include not only traditional licensing out agreements, but also co-development and the establishment of joint ventures. We believe that such partnerships do not necessarily require full clinical completion or proof of concept, and that a potential transaction involving OliX 301A could materialize at various stages during clinical development. Moving on to one of the most advanced areas under our OliX 2.0 strategy, I will now discuss OASIS Adipose, our adipose tissue targeting platform. OliX 501A is a program targeting obesity and metabolic diseases, for which we have generated preclinical data in both normal and obese non-human primates. The target of this program is ALK7, a well-established gene, where suppression of its expression has been shown to promote lipolysis. Further details will be presented in the research session shortly. However, to briefly highlight, in obese non-human primate studies, we observed a reduction in visceral fat that was significantly superior to that of global competitors currently in clinical development. Based on these results, we believe this program has strong potential to become a best-in-class therapy. Next, I will discuss OASIS CNS, our platform for CNS disorders. At present, this program remains at the platform building stage, but we are developing a system designed to deliver siRNA into brain tissue through less invasive administration routes, such as subcutaneous or intravenous injection based on BBB shuttle technology. Today, I would like to share preclinical data generated through our collaboration with Vectoris, a company with proprietary BBB shuttle technology. We are pursuing multiple collaborations with specialized BBB shuttle companies, and in particular, we are working closely with both Vectoris and Key2Brain in the area of transferrin receptor, or TFR, targeting technologies. Among these efforts, I will first highlight the results generated through our collaboration with Vectoris. While the detailed data will be presented later in the research session, the key takeaway is that we observed strong target gene silencing across multiple brain regions following either subcutaneous or intravenous administration. Based on our assessment, these results appear to be highly competitive relative to peer data disclosed in the industry. Turning now to our dual platform technologies, I will first discuss the OASIS D platform. OASIS D is a technology in which two siRNAs are linked together and then conjugated with a tissue-specific ligand. As shown on the slide, one example uses GalNAc for liver delivery. With this approach, two different genes can be simultaneously silenced within the same organ. We have successfully established the OASIS D platform, securing a technology that enables the simultaneous suppression of two target genes within a single organ using one therapeutic agent. We have already confirmed strong efficacy of this platform, not only in liver tissue, but also in preclinical ophthalmology studies. Going forward, we plan to expand this platform to nearly all target organs within our development scope. OASIS Duo consists of two siRNA units connected by a cleavable linker in the middle. Each siRNA can be conjugated with a different ligand, such as a lipid or GalNAc, allowing delivery to different organs. For example, one side may be equipped with GalNAc for liver delivery, while the other is conjugated with a lipid for adipose tissue delivery. Once administered, the linker is rapidly cleaved in the body, enabling one siRNA to travel to the liver and the other to adipose tissue, where each can suppress its respective target gene simultaneously. OASIS Duo is an innovative platform that enables a single therapeutic to reach two organs simultaneously and regulate target genes in each organ. We believe this could become a highly important technology for treating complex systemic diseases involving multiple organs, such as obesity and diabetes, and may help reshape future drug development paradigms beyond the limits of single-organ treatment. OASIS Duo is also a highly novel approach for which comparable precedents remain limited in the global industry. We believe this platform could serve as an important bridge toward developing first-in-class therapies at OliX. With that, I will conclude my summary of OliX's recent achievements. Thank you.